Thanks, Scott. So we're going to move to a series of speakers who are going to speak to us about breakthroughs in material science, nanotechnology, and imaging. And our first speaker is Stephen Cramer, who is the Walker Professor of Chemical and Biological Engineering at Rensselaer Polytechnic Institute. Steve is currently conducting research in numerous areas related to protein surface interactions, including prediction of protein binding affinity and multiscale modeling of chromatographic systems, design of chemically selective displacers, and the development of efficient antibody separation systems. Professor Cramer is the recipient of numerous awards, including a Presidential Young Investigator Award from the National Science Foundation. He is an elected fellow of the American Institute of Chemical Engineers, the American Chemical Society, and the American Institute for Medical and Biological Engineering. Professor Cramer has published over 160 papers in peer-reviewed journals, has 9 patents, and has produced 39 PhD students who have gone on to leadership positions in biotechnology and academia. Please join me in welcoming Stephen Cramer.
Great. Well, thank you very much. It's a great pleasure to be here. So this morning I'm going to tell you a story about a big collaborative project I'm involved in that's funded by DARPA to create new paradigm for bioprocessing. So essentially, I go there, right? So first of all, let me acknowledge my collaborators. This entire team is led by Professor Chris Love from MIT, and it's a collaborative effort between MIT, Northeastern, Paul Corporation, Rensselaer, where I work, Latham Pharmaceuticals, and DARPA. And of course, this is a list of the entire team, so I'm just the messenger. So the current paradigm for bioprocessing is huge capital cost investments, extremely localized biomanufacturing. And the issue is, how do you produce medicines cheaper and locally, right? So what this talk is all going to be about, and what this project is all about, is how to essentially use the latest technology in nanotechnology, in genetics, in bioprocessing, and how to integrate that in a certain way where we essentially can have well-controlled, high product quality biologics anywhere in the world at a relatively inexpensive cost. So we want to replace this with a new paradigm. Okay, so this was motivated by a DARPA-funded project, and the biomod requirements are the following. We essentially want to go from the patient directly to the design, right? So the idea of this whole thing is we want to enable novel, flexible methodologies for genetic engineering and modification of microbial strains and cell-free systems to synthesize multiple and wide-ranging biologics. We want a system, essentially a box or a little room, that will produce many biologics locally anywhere in the world, right? To do that, we need a lot of technology development. So this is a shift of the previous talk. This is more about biomanufacturing innovation. And so the idea is we want to develop flexible, portable device platforms for manufacturing multiple biologics with high purity, efficacy, and potency at the point of care, right? So we need to essentially create all of the nanoscale analytics to prove that the product quality is sufficiently high, and we need to do this in a real-time controlled fashion. And the idea is we want to have an end-to-end manufacturing chain, including the downstream bioprocessing, which is what my lab is involved in a continuous flow microfluidics platform. And finally, we want to do this with currently approved biologics. So the idea is not to produce brand new therapeutics, but to take the large number of biologics already developed or that are becoming biogenerics or biobetters and figure out a way to do that in a much more economical and local fashion. So, so again, what are the benefits and risks to the patient? So the benefits are the availability, just-in-time target populations, so we could produce biologics anywhere in the world where it's required in a very rapid fashion. Broad access, not constrained again by location, and other synergies. And of course, there are risks. This involves a lot of new technologies, so we have to also design a system for a multi-product platform. And there's a need to develop brand new technologies from genetics, nanoscale analytics, and separations technology. And of course, there's also regulatory issues, right? For example, this is a totally new paradigm for producing manufactured products. So how do we deal with the regulatory constraints? So, so here is the schematic of the system that we're working on. And basically, we have essentially several components. The first component is essentially the upstream side over here. So one little pointer, right? And this involves all the genetics to essentially create the organisms that will produce this in a rapid fashion, and all the bioreactor design to do this in a very rapid, continuous, and controllable fashion. We then move from the upstream to the downstream, and downstream means biological purification. And in this case, we're developing a new paradigm for purifying biologics based on the monoclonal antibody approach. monoclonal antibodies are produced now in the companies with a protein A affinity adsorption step, followed by several polishing steps. And this has worked extremely well, and in fact has been responsible for some of the huge successes in producing monoclonal antibodies. But for other biologics, this paradigm does not exist. So the idea here is to create affinity capture reagents, and then to use state-of-the-art multimodal adsorptive systems, and to have essentially a generic platform for producing all of these biologics. Once we purify them, we have to essentially do the final formulation with ultrafiltration, diafiltration, right? And then, of course, we then have all the analytics, right? So the goal now is how do we do online nanoscale analytics to look at product quality as you're doing the process and control the entire process. And then in addition to that, how do you use the analytics to prove that the product has sufficient quality for release, for product release? So, and when you put all this together, how do you control the entire thing? So we're using state-of-the-art control algorithms from chemical engineering to essentially control this entire process and to enable us to constantly improve the quality of the product. So this is a new paradigm for biological manufacturing. So some details. We'll first talk about the upstream side, right? So the organism we're using here is Pichia pastoris, and the advantages of using this organism are many. First of all, it's now actually been used for a late-stage biological development for interferon. It has reduced risk of viral contamination and has human-like post-translational modifications. It's a very genetically stable organism. It has high-density cultures, and it has a high yield of secreted proteins, limited host cell protein background, and it's very amenable to lyophilization. So the idea is we would essentially genetically engineer the Pichia to make a certain biological product, or to be turned on and off to make different biological products. We would then essentially lyophilize it, and it would essentially be in a container. And then when it was brought to the remote location, it would then be brought back into solution, already at a high cell density, and basically be ready to go to produce the biological product, right? And so the advantage of this, again, is it has programmable synthetic circuits for multiplex toggling of genetic pathways. The Love Lab has come up with a very clever way of doing high-throughput clonal analysis and selection. And we also have these rapid bioreactors. So essentially, this all comes together to very quickly be able to develop the specific genetics for using Pichia for making a certain biological product. We then go to the actual reactor. So the reactor itself is a perfusion reactor. It comes out of a company out of MIT called Ferrix. And essentially, this is a microfluidic-based platform for producing biologics very quickly with very precise control of all the key ingredients. The next step in the process is the downstream processing. So for those of you who don't know, whenever you produce a biological product, the purification is an extremely important part of producing this biologic. In fact, the cost can be up to 70 to 80% of the cost of the biological. And that is a really big deal, because if we can decrease the cost of purification, we then, of course, radically reduce the cost of biological products. And again, the paradigm we're trying to do here is have a generic affinity capture and then multimodal bind dilute flow-through operations to purify all of these biologics. So the first step of this is the affinity capture. And the work that's going on at RPI in my lab and Professor Karandi's lab is essentially to design unique peptide affinity reagents for selectively capturing and eluting the biological product. And we do this in a rather clever way. So the first thing we do is we do a combination of high-throughput screening and also rational design. So we look at the epitope mapping of natural binding partners. We look at the affinity ligands of homologous or structurally similar proteins. And then we use, of course, surface mapping techniques, de novo design, and of course, we use phage display. So we employ these different techniques, and we then go ahead and we make them on a nanoscale synthesis screening device. And again, we have various strategies for optimizing these peptide affinity reagents, and I won't get into that in detail right now. So once we do this, we first of all have the screening at the nanoscale, and I'll show you a system for that in a minute. The beauty of that system is that everything is synthesized in addressable spots, and you can then screen it directly. And in that one location, you can very quickly get information about all the things you want to know right at the nanoscale, right? And what we do mainly is we screen at the nanoscale for affinity and for selectivity. Once we have the lead compounds, we then go to the resins. So these are porous chromatographic resins. We also synthesize those in a high-throughput fashion and evaluate those. And then we do this in an iterative fashion to quickly come up with a affinity capture step for a certain biological product. So the peptide synthesis screening technique is carried out in the following way. Essentially, we have, again, these addressable spots where we grow the peptides. And then in that location, we then do the analysis to evaluate their efficacy. And there we go. There's some spots. And what we do is we essentially look at, we want to have high affinity and high selectivity. So it's not just enough to have a high binder, but you need to have a high binder which will also not bind the other products. And the key question is, what are the other products? So again, in Picky, the advantage is that HCPs are at relatively low levels, but you always have product-related variants. So the key thing is, what are you screening for, right? So we basically developed technologies for developing the affinity capture reagents. But we also, when we make these peptides, we've also developed affinity peptides that can be employed in the analytics, both for product quality and for product concentration, right? And again, once we make it at the resin scale, we essentially do this in a high-throughput robotic fashion. And we essentially look for affinity, elutability, selectivity. These are concepts that are important for bioprocessing. And we essentially use this in an iterative fashion to relatively quickly come up with a new affinity reagent for a certain biological product. And we also can do the screening for the elution, so we can essentially find what conditions will selectively elute our product in a very high purity, and how will that interface ideally with the subsequent polishing steps. So we want to have an integrated process everywhere from the upstream to the downstream to the analytics, and have all the sub-processes within the downstream process be well integrated. So this is just a UPLC trace of a recent result from our lab a couple of weeks ago. And basically, we see in the black, we have the feed, right, of the reverse phase chromatogram, and in the blue, we have the product, right? So these things are very effective at getting very high purity in one single step. And then we couple those to a polishing process. So once we've captured it and gotten rid of most of the impurities, we now have to get it to the very, very high level of purity. And for that, we're working with Pall Corporation, which has done a really excellent job of developing new technologies of multimodal resins and membrane systems. And we work together very nicely to integrate the entire process, right? And of course, they do everything as well in robotic high-throughput screening. It's a big movement in the industry. Now every company will have these TCAN robotic systems, and they'll do their methods development for biomanufacturing at the robotic scale to rapidly increase the efficacy and decrease the time for methods development. So here we see some examples of getting rid of DNA and getting rid of host cell proteins. And the bottom line is, when we do this, we can get extremely high overall purity with very high yields, and the results are as good as the commercial standards. So now we go to the next step. Once we've purified this, we now need to formulate it in the final condition which will be employed for administering the drug. And for this, Pall Corporation has come up with a scaled-down UFDF ultrafiltration diafiltration membrane system, and the system has worked extremely well at very small scales at purifying what we want. So it's interesting. We've been focusing on the scale down of the system, right, for proof of concept at the order of like a couple of doses. But the scale up actually would be easy, right? So the scale down was the challenge. The original dictate from DARPA was to make this as sort of like a backpack that a soldier could wear into the field and essentially make drugs anywhere where they were. And of course, that's the goal of what we're doing. But the technology itself is very amenable to scale up. So instead of a backpack, it can be a room, right? And it can deal with orphan drugs and a variety of other indications. And finally, we move to the release analytics. And so this is the part that's actually, I think, almost the most exciting, in addition to the genetics part. And this is essentially using state-of-the-art nanoscale analytics for online sensing of the product quality, right? And so what we have here is we have the online on one side and the offline on the other side. The offline are state-of-the-art, large-scale offline apparatus that people employ, right? Typically LCMS, different ELISA, Biocore, standard technologies. And on the other side, we have the new technologies. So the carbon nanotube-based sensors, the radically scaled-down Raman spectroscopy, the nanofluidics for purifying for analyzing the proteins based on size or electrokinetic behavior, and of course, product quality, activity, safety. This is all being done at nanoscale in real time inline, right? At least that's our intention. So here we have the old school, if you will. This is the offline analytical techniques, and we see how this is typically done in the biomanufacturing industry. We essentially have, again, we have the ELISAs, we have the Biocore, the LC mass spec, and then what does the new technology look like? So essentially, one of them is a miniaturized Raman system. So I'm sure you've all seen these Raman systems that are huge. And the idea here is to scale this down to a very small scale that'll then be able to deal with very low concentrations. So we want to scale it down and increase the sensitivity. And this is work out of the Raman lab that's done some really remarkable work at developing scaled-down Raman systems. Another technology is the nanofluidic size-based separation. This is based on essentially a very clever design of these nanoscale systems to very precisely get size-based separations very quickly, right? We also have product quality with nanotube sensors. We can distinguish glycosylated versus non-glycosylated products. We can also distinguish properly folded versus unfolded proteins. And what we do is we then bring this all together now for a new paradigm for process analytics. So we have a new way of doing the upstream genetics, a new way of doing the purification, and then a new way of doing essentially the analytics. And then what brings it all together, of course, is the control. So we then have precise mathematical models of each one of these unit operations, and we bring that together in a very precisely controlled manner with the right sensors so we can do online control of the process to always keep the product quality extremely high, right? And then also to prove the final product quality for release. And so, next slide, right? So this is actually, doesn't look so fancy, but we actually built this thing. So this is actually a scaled-down system that we've actually built at MIT, and we've essentially done proof of concept for this for a certain biologic, and we've actually shown that we can actually do this. And so, just going over here. So again, and one last thing is we have very precise new control algorithms that we're employing directly on this apparatus that we've actually synthesized at MIT, right? So what have we learned so far? First of all, that rapid, high-quality biomanufacturing is feasible, that systems integration and control will be critical for controlling the performance, that a multi-product facility will require extensive process validation, but we believe we can do that, and that redundancy and orthogonality on analytics for real-time release will be very essential. Right? So I just want to, again, acknowledge Professor Chris Love, who's leading this whole effort. I'm leading the effort on the downstream processing, and DARPA for funding, and I couldn't resist one slide on research in my lab. Since I've never been here before, it's a pleasure to have that opportunity. And so my research involves state-of-the-art protein separations, biophysics of protein interactions, multi-scale modeling, smart biopolymer systems, lab-on-chip devices, and a variety of basically separations technologies. So I look forward to spending the next 2 days at this conference and meeting many of you and looking at opportunities for potential collaboration. Thank you.